un letrero.
Ya empezó.
Welcome everyone. It's a pleasure to have Stuart Kauffman with us today. And well, he doesn't need much of an introduction. He has been a founding figure in the scientific study of complex systems, working several years at the Santa Fe Institute and more recently at the Institute of Systems Biology. And many of you are already aware of his contributions. So today he will speak about Schrodinger's what is life and what can we learn from it. Stuart, please, we're eager to listen what you have to say.
Thank you, Carlos. And I'm glad to have the chance to talk. There's something good about doing things by Zoom. You can't talk to one another afterwards, but it's got its virtues. I gave this same talk 2 weeks ago at the Karolinska Institute in Stockholm. Can you see the first slide, Carlos? Carlos?
Yes.
Okay, so you can see the first slide. So my title was "What is Life? Answering Schrodinger," which is already grandiose. And Ingemar Ehrenberg, my dear friend, changed the title to "What is Life? The Future of Biology," which is even more grandiose. So I hope you'll forgive it. So I'm not going to talk about Boolean nets. I'm going to try to talk about what Schrodinger had to say, and it leads us in lots of interesting directions. So here it is. So that's slide 1. I guess I can do that. So I have to read this. So I will be reading to you what I write. My purpose in this lecture is to discuss quantum physicist Erwin Schrödinger's superb book, What is Life?, published in 1944. The answer he gives in the very large problem with which he leaves us. I will put this in the larger framework of the origin and evolution of life on Earth from 3.7 billion years ago and the vast evolutionary flowering of complexity in the biosphere. Or as Darwin said at the end of The Origins of Species, from so simple a beginning. Let's see if this works. So here's some references. I'll just say them. If you've not read Schrödinger's What is Life, do read it. Another interesting book is Complexity and the Arrow of Time, Lineweaver et al. Paul Davies has written a very good book called The Demon in the Machine about life. He wants to tie the demon to, of course, Maxwell's demon. I've written a book that came out a year ago, A World Beyond Physics, The Emergence and Evolution of Life by me. I will be able to get to a lot of this today, but not to the fundamental point of my last book, which is there's no law whatsoever that entails the becoming of biospheres. I will not be able to talk about that today. If you would like to find that, if you go to the journal Angelaki, Angelaki, in June, a dedicated volume came out, and I have an article called Eros and Logos about that topic, and my last book covers it. David Deamer, Assembling Life, and as we'll see, Autocatalytic Networks of the Origin of Metabolism by John and Xavier et al., Proc. Roy Sock B, March 2020. I'll talk a bit about Joanna's paper. In What is Life, Schrödinger does not ask what life is, or how life emerged from non-life and evolved. I'll make some proposals below. Schrödinger asked 2 major questions. 1, what is the source of order in organisms? 2, how do organisms remain ordered in face of the second law of thermodynamics? I'm going to propose that Schrödinger's answer to his first question, his famous aperiodic solid, is also the still unarticulated answer to his second question. Excuse me while I try to adjust some light here. Schrödinger notes towards the aperiodic solid, Schrodinger notes that there is a source of order from disorder seen in statistical mechanics. For example, the diffusion of a drop of blood in a coffee cup to a uniform mixture at equilibrium. Diffusion yields order from disorder. He notes that the basis of this macroscopic order is the chaos in the random motion of the atoms. Then Schrodinger notes that as the number of molecules in the system is reduced, the system becomes more random. Macroscopic order from disorder depends upon a large number of atoms. He next considers recent evidence, it was really recent at that time, from the X-ray induction of heritable mutations, that the hereditary material genes can have only on the order of a few thousand atoms. Thus, order from disorder cannot explain the order in organisms. He concludes that the order in organisms must be due to quantum mechanically stable molecules, which are stable despite having a small number of atoms. So that's absolutely sensible. His next move is what's brilliant. Schrodinger then brilliantly concludes that the solid will not be periodic like quartz, for these are, as he says, dull. Rather, the hereditary material will be an aperiodic crystal that contains in some kind of code script the entire pattern of the individual's future development. The code script will be efficacious in bringing about the organism. Here's his quote. In physics, we have dealt hitherto only with periodic crystals. To a humble physicist's mind, these are very interesting and complicated objects. They constitute one of the most fascinating and complex material structures by which inanimate nature puzzles his wits. Yet compared with an aperiodic crystal, they were rather plain and dull. So here's the crux point. The difference in structure is of the same kind as that between ordinary wallpaper, in which the same pattern is repeated over again and again in regular periodicity, and a masterpiece of embroidery, say a Raphael tapestry, in which shows no dull repetition, but an elaborate, coherent, meaningful design traced by the great master. Pause for a moment here. Here's Schrodinger, and he's trying to think about biology. This isn't any specific molecule. This is the intuition that whatever the source of information is going to be in biology, it can't be dull. This is brilliant step one. And it's how you do science and do a lot of great science. He just did it. It is these chromosomes that contain in some kind of code script the entire pattern of the individual's future development and its functioning in the mature state. Every complete set of chromosomes contains the full code. It is Schrodinger's brilliant intuition of an aperiodic solid that contains, quotes, in some kind of code script the entire pattern, that inspired Watson and Crick in their 1953 discovery of the structure of DNA, which Avery had shown to be the genetic material a few years earlier. DNA is precisely an aperiodic solid. In the next decades, the existence of the genetic code was deduced and worked out. Thus, Schrodinger concludes the order in organisms does not come by order from disorder, as in diffusion, but order from order. Schrodinger's second question, how do organisms remain ordered in the face of the second law of thermodynamics? Here's his words. How would we express, in terms of the statistical theory, the marvelous faculty of a living organism by which it delays the decay to thermodynamical equilibrium, or death? The device by which an organism maintains itself stationary at a fairly high level of orderliness really consists in continuously sucking orderliness from its environment. Schrodinger's use of the phrase sucking orderliness from its environment is also called negentropy. Critically, Schrodinger does not see how a second question is covered and answered by the known laws of classical and quantum physics, quotes, We must therefore not be discouraged by the difficulty of interpreting life by the ordinary laws of physics, for that is just what is to be expected from the knowledge we have gained of the structure of living matter. We must also be prepared to find a new type of physical law prevailing, or are we determined a non-physical, that is to say, a supraphysical law? In short, Schrodinger doesn't know the answer to his second question. So I'm going to try to provide an answer to his second question. I think I get at least part of it. The rest of us can all think about it, and it may be on the right track. I'm going to start with non-equilibrium dissipative structures that I think you all know about. Part of the answer to Schrodinger's second question is already contained in his phrase, really consists in continuously sucking orderliness from its environment. In systems displaced from equilibrium by the flow of matter and energy through them, macroscopic patterns can emerge. Dissipative structures were unknown when Schrodinger wrote in 1944. Ilya Prigogine and his group, largely in Brussels, led a large effort to study the onset of ordered patterns in far from equilibrium physical and chemical systems, and termed such patterns dissipative structure because they're dissipating energy. Examples include whirlpools, tornadoes, hurricanes, Bénard cells, and the Belousov-Jabotinsky reaction. The giant red spot on Jupiter is an example. In these systems, the macroscopic pattern emerges as matter flows through the system, which is therefore displaced from equilibrium by that energy flow. The Bénard cell is an instructive example. Here, there is a pan with a shallow layer of viscous liquid, for example, oil. The pan is heated slowly from below, creating a temperature gradient, hotter on the bottom than on the top of the fluid. When the temperature gradient induces an overall heat flow to the environment. When the gradient is small, the heat flow to the environment is achieved by conduction by the random motion of oil molecules. When the temperature gradient is above the Rayleigh threshold, convective cells arise and dissipate heat more efficiently. The convective cells are Bénard cells. Bénard cells patterns are sustained by the continuous flow of energy through the system by heating the pan from below. So I'm going to take a next critical step. The form of these cells depends upon the boundary conditions created by the shape of the pan. The pan can be a circle, it can be a triangle, it can be a rectangle, it can be any shape you want. The Bénard cells can be hexagonal or rolls. Here's the critical point. If the shape of the pan, that is to say, the boundary conditions are changed, so too are the shape and the overall pattern of the Bénard cells. So I'm moving in a first step towards answering Schrodinger's second question. I propose below that Schrodinger's aperiodic solids that constitute a code script that is effective for the formation of the organism is because the aperiodic solid is one or a set of boundary conditions that thereby control the dynamics of the non-equilibrium system in living cells. Thus, as we will see in more detail below, Schrodinger's answer to his first question is the answer to his second question. So we're going to be going through different pathways getting there. So this looks like a side trip, but it's not quite. Dissipative structures such as hurricanes, whirlpools, and Bénard cells do not become very complex and increasingly diversified in their complexity. By contrast, the evolution of life started with rather simple protocells, followed by single-cell organisms such as archaea and eubacteria, then eukaryotes and multicell organisms, and the Cambrian explosion 550 million years ago with the flowering of some 13 extant phyla to millions of distinct species today, bacteria to whales, and redwood trees in an open-ended evolution. By the way, is really important, we do not understand the conditions for open-ended evolution. Algorithmic systems so far have not done it. That's a side comment. What is the difference between dissipative structures and evolving life? Part of the reason that dissipative structures do not become indefinitely complex, and this is the essential point, is that they do not construct their own boundary conditions. Organisms, as we shall see, do construct their own boundary conditions and do this by carrying out thermodynamic work to construct those very boundary conditions. These are quite new things. Okay, work, constraints, and the work constraint cycle. Work is force acting through a distance, Newton. So I accelerate a hockey puck in some direction. I've done work on it. Then Atkins in the second law says, work is something much more interesting than that. Work is the constrained release of energy into a few degrees of freedom. This is going to be essential. Work is the constrained release of energy into a few degrees of freedom. So let's consider a cannon and a cannonball inside the cannon. The explosion of the powder releases energy into the few degrees of freedom left as created by the cannon itself, which constrains the release of energy into those few degrees of freedom, so the cannonball can only move in the direction of the cannon shaft. Physicists mean by degrees of freedom one of the possibilities. So if there weren't a cannon there, as we'll see, the powder could explode in lots of directions. If the cannon is there, the explosion can only go in a limited number of directions. So here it is. Thermodynamic work is done on the cannonball. The powder explosion is exergonic. The resulting movement of the accelerated cannonball is endergonic. When the powder explodes, part of the released energy is dissipated as heat, producing entropy. But part of the released energy does thermodynamic work to propel the cannonball. So, next point. Critically, were the cannon not present and the powder exploded on a flat plate next to the cannonball, the ball would hardly move and most of the energy would be dissipated rapidly as heat, thereby rapidly producing entropy, and less work would be done. Thus, the constrained release of energy that does work in a linked exergonic endergonic process produces work and at the same time delays the production of entropy longer than were the constraints not there. Begin to recall Schrodinger's statement, delays falling to thermodynamic equilibrium, which is death. I'm going to be making the claim that the constrained release of energy releases the production and release of entropy, and that is a major part of the answer to Schrodinger. Again, when the cannonball is blasted from the cannon, more work is done, and the total production of entropy is delayed. This is going to be fundamental. When more work is done in the constrained release of energy in a non-equilibrium process, the production of entropy is delayed than in the process than were the process less constrained. This is going to be a central part of the answer to Schrodinger's second question. Macroscopic propagating thermodynamic work. In an automobile, the explosion of gas causes the piston to move, which causes the crankshaft to turn, which causes the wheels to turn. Thermodynamic work propagates in a coupled set of exergonic and endergonic processes from the gas explosion to the wheels turning. This is propagating work. A large number of macroscopic changes happen in the world. The cannon, which constitutes the constraint on the release of energy into a few degrees of freedom, is a boundary condition. Similarly, in an automobile, the cylinders and pistons, gears and escapements are boundary condition constraints on the release of energy from the exploding gas into a few degrees of freedom that constitutes propagating macroscopic work by which the engine leads to the physical motion of the car on the road. But that raises a very interesting question for the cannon and whatever else. Where, since the Big Bang, did the cannon or cylinder piston boundary conditions come from? There were no cannons around 13 billion years ago. After the Big Bang, thermodynamic work was required to construct the cannon, the cylinders, the pistons, and the escapements. And this leads to what I will call the work constraint cycle. No constraints on the release of energy in a non-equilibrium process, no work. But also no work, typically no constraints. Very often, work must be done to construct constraints. We haven't thought about that, but it's going to be fundamental. In our human machines to date, we organize the work to construct structures, often low energy stable structures such as cannons and pistons, that are then used as boundary condition constraints on the release of energy into a few degrees of freedom, such that propagating thermodynamic work is done. Engines turn over, the car moves. So again, since the Big Bang, where did the boundary conditions come from? We will see below that cells and organisms do thermodynamic work to construct their own boundary conditions. Automobiles do not construct their own boundary conditions. We're moving in on something central that's different in life than non-life, in addition, of course, to reproduction. So I now want to talk about boundary conditions. In classical physics, the boundary conditions can vary freely, so can carry a causally effective code script. Newton's framework for classical physics are his law of universal gravitation and his three laws of motion, coupling the variables in differential equation form, plus initial and boundary conditions, which are not themselves, the initial and boundary conditions are not specified in the laws of motion. Using the initial and boundary conditions, we can integrate the differential equation laws of motion and obtain the entailed trajectories of the balls, the variables, for example, the billiard balls on a billiard table. The boundary conditions determine what the phase space of the system is. For example, on a billiard table, the boundary conditions given by the table's edges or boundaries thereby specify all possible positions and momenta of the balls on the table. But the set of all possible positions and momenta constitutes the phase space of the system. So if you keep the same boundary conditions and alter the initial conditions, the trajectories of the balls are in the same phase space, like billiard balls on the same table. But if you alter the boundary conditions, for example, the shape of the billiard table, that alters the very phase space of the system. But Newton's laws leave free what the boundary conditions might be. So I'm going to talk about boundary conditions. Now, in the United States, we have what are called Pee-wee golf courses that you take your 8-year-old children to. So I was on a Pee-wee golf course recently. It consisted of 18 golf holes. Each hole was created by arranging 6-inch by 6-inch across and long timbers on the ground in various arrangements. For example, the major timbers were parallel to one another, creating pathways, then turned corners at various angles. Short timbers were arranged jutting out into the pathways the parallel boards created. The golf ball, once hit, caromed or bounced off the jutting short timbers in complex ways. The short timbers and also the long timbers are boundary conditions. If the short timbers are rearranged, the motion of the golf ball after it is hit will be changed. But we are free to rearrange the short timber boundary conditions in many ways. So here's a big step. For such a timber arrangement to carry information, it is essential that the timbers could, counterfactually, have been located in a different way. Counterfactually means contrary to fact. They're this way, but they could have been different. This counterfactual captures the idea that we are free to rearrange the short timbers. Rearranging the timbers in some arbitrary way alters the phase space of the system in some arbitrary way. Pre-summary. Rearranging the timbers in some arbitrary way alters the phase space of the system in some arbitrary way. So we're about to see that that's what happens in organisms. In evolving organisms with DNA, RNA, and encoded protein synthesis, these molecules are all aperiodic solids, and they constitute boundary conditions on the constrained release of energy in myriad enzyme-catalyzed chemical reactions into a few degrees of freedom that does thermodynamic work to construct further and often changing aperiodic enzymes that are further boundary conditions.
Kauffman, estoy diciendo que es la vida.
Sí, ya sé, mira.
Pero no dice mucho.
Could some people please mute their system, please?
Oye, me gusta. What is life?
What is life?
Could people make sure their systems are muted? I don't mean to be obnoxious, but please do. Okay. In cells, thermodynamic work is done
No
to
tienes que comer
synthesize
cosechera
RNA
porque
and
está
protein.
limitado.
Sí, me diga.
Entonces, ahí está la leche. Digo, no sé qué tan cargado quedó en realidad.
Rico.
¿Sí? Porque no lo probé.
Sí, neni. ¿Cómo han seguido hoy tus padres? Bueno.
Per piacere non parlare.
Sí, estaban mejorcitos, sí.
Carlos, did you do anything?
I already wrote to him.
Okay, that's fine. Okay. In cells, thermodynamic work is done to synthesize RNA and proteins. The new RNA and protein sequences are new boundary conditions, and their construction persistently changes the very phase space of the cell and the organism. So let's look at it in particular. In cells, like us, we have genetic regulatory network that turn genes on and off. So different cell types are making different combinations of proteins, each of which is a boundary condition. So different cell types expressing different proteins that are boundary conditions. It's strange to think this, but they're living in different phase spaces. It's just like rearranging the timbers for the PB golf course. In single cell organisms over the life cycle, the RNA and protein present change. These changing RNA and protein sequences contains causally effective information that creates different phase spaces. I'm going to say that boundary conditions contain causally effective information. So that's a central theme here. This causally effective information is Schrodinger's causally effective code script. So let's proceed more slowly. The critical step I now take is to identify Schrodinger's code script, to which Schrodinger points, as the very boundary conditions constituted by the aperiodicities in the aperiodic solids in a living cell or system. So let's just think about a specific example of the hereditary DNA molecule with, in this case, the aperiodic sequence of nucleotide bases A, T, C, and G. So the sequence of bases A, T, C, and G in the DNA are indeed boundary conditions. DNA is transcribed into RNA, in the simplest case, into a messenger RNA, A, U, C, and G. That RNA sequence is loaded with 20 different amino acids by specific transfer RNAs, each of which binds to a specific amino acid at its amino acid binding site and binds to the RNA sequence codon via the transfer RNA's anticodon site. Specific encoded synthetases and antibodies attach the proper amino acid to its proper transfer RNA. As the RNA is so loaded, it is processed by the ribosomes to link the amino acids into a growing peptide by forming peptide bonds. Work is done at each step, utilizing the degradation of ATP to ADP as new bonds are formed. At all steps, one or more molecules act as boundary conditions on the reaction occurring. For example, the single-stranded DNA molecule uses Watson-Crick bonding to bind and line up adjacent free AUCG nucleotides of the growing RNA molecule. By so binding these free RNA nucleosides type bases, their three-dimensional freedom to diffuse is confined to one dimension, thereby lowering the activation barriers to form the proper 3', 5' phosphodiester bond between adjacent RNA nucleotides. In forming those bonds, thermodynamic work is done using ATP degradation to ADP as the energy source, and a new chemical bond is formed. In short, the single-stranded DNA acts as a boundary condition, quotes, enzyme, unquotes, binding the substrate RNA bases and lowering the activation barrier, thus enabling chemical energy of ATP to be released into a few degrees of freedom, thus forming the specific 3', 5' phosphodiester bonds between the two RNA nucleosides, rather than the thermally more favorable 2', 5' bond. So in general, enzymes bind their substrates, one or more substrates, and thereby they reduce the motion of the substrates in 3D to 1D or 0D, and By binding to the transition state of the reaction, they lower the activation barrier and the chemical energy is released in a specific direction to create specific products. So enzymes are, in general, boundary conditions. The RNA, DNA, and encoded proteins carry information because they counterfactually could have been different. So one has to think about this for a while, but I think it's correct. They all could have been different. We can come back to that if you want below. This arises both in evolution by mutation and recombination and in changing patterns of gene expression in single cells during development of multicellular organisms with cell differentiation. In evolution of the sequence of bases, counterfactually could have been different. For ontologically indeterminate quantum mutation event could have happened and, say, converted an A to a G. This mutation freely rearranges the nucleotide sequence on the DNA. The freely rearranged DNA sequence via the ontologically indeterminate mutation constitutes a new boundary condition that encodes information because it is a boundary condition, so causally efficacious and counterfactually could have been different had the mutation not occurred by a random mutation event. I conclude that Schrodinger's code script is causally efficacious and includes the DNA, RNA, and proteins that act causally efficacious at freely changeable boundary conditions. Of course, other features of cells, such as membranes, organelles, microtubules, are also boundary conditions. So, continuing, thus the code script is written in a changeable set of boundary conditions that allow thermodynamic work to be done in catalyzed chemical reactions in different specific ways, even as entropy is created. But if the release of energy is more constrained, more work is done, and the release of entropy, I should say the production and release of entropy is more delayed than were the release of energy less constrained. Life, so where you see release of entropy, it should be production and release. I just haven't fixed it yet. Life, think about this. Life sharply constrains the release of energy to do more work. And so the production and release of entropy is more delayed than otherwise. This is the central part of how life surpasses mere dissipative structures and copes with the second law of thermodynamics. In summary, Schrodinger's answer to his first question, aperiodic solids, answers his second question. So I want to talk a little bit about you see where I say delay in the release of entropy at the top? I should have said production and release of entropy. I just haven't fixed it yet. Consider again the cannon and the cannonball. The cannon boundary conditions yields a constrained release of energy when the powder explodes, thrust the cannonball out the cannon, thus is thermodynamic work. Were the cannon not present, the powder explosion would have released entropy, produced and released entropy more rapidly, and less work would have been done than if the cannon is present. Well, the production and release of entropy is more delayed and more work is done. Everywhere I have release, it should be production and release. Thus, in general, work is the constrained release of energy into a few degrees of freedom. If the constraints increase so that the energy is released into fewer degrees of freedom, more work is done, and the production and release of entropy is more delayed. I'm sorry I don't have the word production in there. Thus, highly constrained release of energy corresponds to delay in the increase in entropy. It's going to be central to the delay in falling to thermodynamic equilibrium, of which Schrodinger speaks. I apologize. This is how, in answer to Schrodinger, life copes with the second law. Highly constrained release of energy to very few degrees of freedom occurs, which is work done in cells, and the production and release of entropy is delayed. That work constructs the, so I'm moving on to a new topic. Once work is done, the work can construct new boundary conditions on the release of energy into few degrees of freedom that constitutes work. I'm bringing us towards a new topic called constraint closures. This is the astonishing thing that automobiles do not construct their own boundary conditions, cells do. So here it is. Some of some of the work done constructs the very same set of boundary conditions that then constrain the release of energy, such the very same work is done. In a moment, we will see that this is constraint closure. It's not my idea. I'll tell you about it in a minute. As these processes occur, each does more work, and the production and release of entropy is more delayed than were the constraints fewer, so less release. So less work is done, and the production and release of energy is more rapid. So again, this is how life copes with the second law to produce a sustained state of order in a volume of space and time in living cells. Again, Schrodinger's answer to his first question is the answer to his second question. Now, I want to get to this beautiful idea of constraint closure. It was invented by Matteo Mosio and Mael Monteville, published a few years ago, 2015. It's a truly brilliant idea. So here it is. We saw above that thermodynamic work can be done to construct constraints, structures. But this means that thermodynamic work can be done to construct structures that are themselves available to be used as boundary conditions, that is, as constraints. The constrained release of energy can construct more constraints. For example, machine tools can construct cylinders that themselves can be used in an automobile as constraints on the release of energy to do the work to power the car. So this is Matteo and Mosio, Constraint Closure in 2015. So we have to go slowly here. It's such a brilliant idea. You'd think you could get it the first time. It's a little subtle. In a constraint closed system, there is a set of non-equilibrium processes. Let's call them 1, 2, and 3. Each is constrained by one of a set of constraints, A, B, C, such that the constrained release of energy in the processes 1, 2, 3 does thermodynamic work to create the very same set of constraints. So work is going to be done to make some constraints, and the constraints are what constrains the non-equilibrium process to do the work that constructs the constraints. It's an utterly brilliant idea. For example, process 1, constrained by A, constructs B. Process 2, constrained by B, constructs C. Process 3, constrained by C, produces A. So that, for the little example that we have above, is precisely a constraint closed system. So some of you may know this old idea. It's actually mine of collectively autocatalytic sets of proteins. I'll get to it in a minute. Gonan Ashkenazi has one in the Ben Gurion. It achieves constraint closure. This constraint closed system is remarkable. It is an open thermodynamic system carrying out a cycle of thermodynamic work, task 1, task 2, task 3, with the formation of entropy whose production and release is delayed. I've got to fix this everywhere. The production and release is delayed. The entire system constructs the very constraints on the release of energy by which the work is done to construct the very same constraints. This is an automobile that constructs itself, okay? Because the release of energy is constrained, more work is done, and the release of the production and release of entropy is more delayed than were the release of energy less constrained. These constraint closed systems, these constraint closed and far from equilibrium processes are organized. So I apologize everywhere for using just the word release. By making a more constrained release of energy, the production of entropy is delayed. It can be delayed for a long time. Also by building stable structures that then break down when you die. So this is a non-mystical holistic closure. It's achieved by the system above. No constraint constructs itself. The set of non-equilibrium processes and constraints mutually construct the joint set of constraints by linking the set of non-equilibrium processes into a closed set of work tasks to be performed jointly. That's constraint closure. So a constraint closed system constructs itself. Okay, so I need to, and don't yet have in this still early outline, a big summary here before we go into a life itself. So the summary of all of this is, so Schrodinger says, we understand the aperiodic crystal, and then Schrodinger says, how do organisms remain stable in the face of the second law? So step one in the answer is that the information is carried in aperiodic solids that constitute boundary conditions like enzymes. The second step is work is the constrained release of energy into a few degrees of freedom. The third step is if the release of energy is more constrained, more work is done, and the production of entropy is more delayed. Step four is a constraint closed system constructs its own constraints on the release of energy and builds itself. Constraint closed systems build themselves. And if they are very good at constraining the release of energy in building themselves, more work is done and the production of entropy is more delayed. So let's talk about what that has to do with thermodynamics. Here's a simple example, and I haven't written this down clearly yet. Suppose a cannon fires a cannonball at noon in Mexico City. And it takes a few hours for the cannon and the cannonball to cool off. Well, when the cannon and the cannonball have cooled off and come to equilibrium with the surrounding temperature, all of the entropy has been produced and released. Now, suppose instead that it's a very cold night in Mexico City, and about an hour and a half before sunset, the cannon fires the cannonball, and they stay warm for quite a long time, and it becomes night. Well, when it becomes night, photons are radiated into space. Because the cannon and the cannonball are still warm, they radiate more energetic photons, bluer photons into space than red photons into space. So the entropy of the planet stays lower than it would have otherwise. And this is a simple example to show that by delaying the production and release of entropy, the system involved can remain stable in the face of the second law, because the produced entropy is delayed and then released elsewhere, for example, into space. This isn't well worked out. This is the start of really understanding it. So this needs a summary here, and that's my roughed-in summary. I'm now going to turn to life itself. We have about, I guess, 20 minutes to go. So Schrodinger never says, how do we ever get life? Well, we've made a lot of progress in the last 50 years or 60 years, or let's see, 84 years since Schrodinger. There are several theories for the origin of life. These include what's called the Rna World and the theory of the spontaneous formation of collectively autocatalytic sets, CAS, made of RNA, of peptides, and of lipids. So Dave Deamer talks about this. Paul Davies does a little bit. I certainly do in my last book. But in fact, if I could brag a little bit as a grumpy old man, it was actually my idea, and I published it in 1971, that life may be the emergence of collectively autocatalytic sets. And I published it in 1986 in a bunch of my books. So what's a collectively autocatalytic set? Well, let's get first to the Rna World. Basically, the Rna World proposes that there's it's entranced with the template couplet matching properties of a double-stranded DNA or RNA. So the idea is you have a long single-stranded RNA, and it is able somehow to template copy itself. So the Watson strand lines up the free nucleotides that will be the Crick strand, and the single-stranded RNA molecules links the free nucleotides to synthesize the Crick strand, and then vice versa, and you have a template replicating RNA molecule. That is at the heart of the Rna World. It has never worked. It might work. The best hope right now is that somebody in England, whose name I have forgotten, has evolved an RNA molecule in vitro that can link together about 200 nucleotides to copy another RNA molecule, and that's superb. There's lots of problems with what I'll call the nude gene point of view. How does it gather around itself a connected metabolism? How does coding get going? Lots of other problems. So the other guy is a collectively autocatalytic set. So that idea is the following. Say there's Carlos and me. We are two peptides. I catalyze the formation of Carlos by gluing together two fragments of Carlos into a second copy of Carlos. Carlos catalyzes the formation of Stu by gluing together two fragments of Stu to a second copy of Stu. That's the simplest possible collectively autocatalytic set. Neither Carlos nor I catalyze our own formation. I get to be reproduced because of Carlos and vice versa. So we're collectively autocatalytic set. We're also what's called a Kantian whole, where the whole exists formed by means of the parts, and the parts exist formed by means of the whole. So autocatalytic systems have actually been made. They were first made of DNA, Gunter von Katarowski in 1994. They were done with peptides in 1995 and more recently. Goran Ashkenazi and the Ben Gurion has a 9-peptide collectively autocatalytic sets. They've almost been done with lipids, and they've been done with RNA. This is a crux point next. Since a set of peptides can collectively produce, molecular reproduction does not require nucleotides. The RNA world may be true, but it's not necessary for life. So I've already just said that. Oh, such collectively autocatalytic sets, for example, peptides and RNA are both, achieve constraint closure. The peptides or RNA constituents are themselves enzymes, which are boundary conditions of the very reactions which makes the same peptide or RNA enzymes are formed. So a collectively autocatalytic set does two magical things. Well, three. It's a non-equilibrium chemical system. It's molecularly reproducing, and it achieves constraint closure. In this process, again, thermodynamic work is done to construct the peptides, which themselves are boundary conditions on the release of energy into a few degrees of freedom. Entropy is produced in this process, but its production and release is more delayed than where there are less constraints on the release of energy. Again, I apologize. Again, this answers Schrodinger's second question. So again, such a collectively autocatalytic set has three closures: constraint closures, work task closures. All the tasks that have to get done. Carlos catalyzes my formation, and I catalyze Carlos' formation. If we call those catalytic tasks, they all get done. And catalytic closure, every task that has to be catalyzed is. The release of energy is delayed. I keep not fixing that. The production and release of entropy is delayed. The set of three closures is a non-mystical holism. Thanks to the three closures, such systems literally do thermodynamic work to construct themselves. It's not just an astonishing thing. The biosphere constructs itself. Redwood trees construct themselves. So I'm going to now tell you new evidence that I think is stunning, that life is widespread in the universe. Xavier et al. I am in fact a co-author on this paper, but I didn't do the work, so I can brag about it. The title of the paper is Autocanonical Chemical Networks at the Origin of Metabolism. Proc Roy Soc B, March 11th this year. Joanna led the whole effort with Bill Martin. Archaea and bacteria, from before oxygen was in the atmosphere a couple billion years ago, have collectively autocatalytic sets formed of about 1,500 small organics and organometallic compounds with no polymers of DNA, RNA, or protein. Almost certainly, this is the new thought, almost certainly molecular reproduction started with such sets of organics, but no polymers. In short, molecular reproduction arose probably before DNA, RNA, and genes and proteins. So the Murchison meteorite, formed 5 billion years ago, has about 60,000 organic molecule diversity. So does Enceladus, a moon of Saturn. So that means that 5 billion years ago, everywhere in the universe, among the 10 to the 22 solar systems, high molecular diversity was present. If collectively autocatalysts can form easily in such rich chemical mixtures, which needs to be tested, the theory says yes. We need to do the experiment. Molecular reproduction started 5 billion years ago on many planets among the 10 to the 22 solar systems. I just think that's amazing. I think Jonah led the work I think is transformative in the origin of life story. So now we've got some molecular reproduction. So now we have to get to protocells. There are several theories exist which house such collectively autocatalytic molecular systems, or RNA. For example, liposomes that divide and create protocells. For example, Roberto Serres talked about it, and Damer and Deemer recently are talking about it. Where they're formed of DNA, RNA, or proteins and linked to a metabolism, all these are aperiodic solids with such as enzymes can act as boundary conditions in non-equilibrium systems on the release of energy into a few degrees of freedom to do thermodynamic work with a simultaneously delay in the production and release of entropy than where the constraint's not there. Such systems can exhibit heritable variation, carrying a code script that is causally efficacious. So now I want to talk about open-ended evolution, and we're doing okay. We have another 15 minutes. So this is an odd thought. The universe is not ergodic. It's not ergodic above the level of atoms. Most complex things will never exist. So here's the way to see it. A typical protein is 300 amino acids in length. So let's consider all possible proteins length 200. Well, there's 20 amino acids standard. So there's 20 to the 200th possible proteins length 200. That's 10 to the 260th possible proteins. Now, could they all be made in the age of the universe? Well, so it turns out, well, let me just do the calculation. There's 10 to the 80th protons in the, particles in the universe. The shortest time scale is 10 to the minus 43rd seconds. If the 10 to the 80th particles were doing nothing but making proteins length 200 in parallel, ignoring space-like separation, in parallel for the 10 to the 17th seconds of the universe, if you calculate it, it would take the age of the universe times 10 to the 37th power to create all possible proteins length 200 once. This means something astonishing. The universe has explored a tiny, tiny, tiny subset of the possible proteins length 200. That's what I mean by non-ergodic. Thus, there's an infinite sink upward in complexity that evolution can explore. So here's the heart of a chunk of the heart of my last book. I'm working on a new paper with Lee Smolin, Marina Cortez, and Andrew Little about this. Human hearts are very complex things, but they exist in the non-ergodic universe. Well, how? Well, living cells are Kantian wholes whose parts exist in the non-ergodic universe by means of the whole. So you exist for and by means of your heart, and your heart exists for and by means of you. When you propagate your kids, you carry along the parts that sustain you, so hearts propagate in the biosphere. Wholes evolve and carry along their sustaining parts. The selection acts at the level of the whole organisms, so hearts evolved a long time ago in the vertebrate lineage and other lineages. They've been carried along. Hearts have been carried along with the whole, namely the whole vertebrate, because selection acts on the whole, but the whole won't stay alive unless it has the functional part. That's why there are hearts in the universe. Life undergoes an ever open-ended evolution. Protocells give rise to bacteria and archaea, to eukaryotes, to sex, to multicell organisms, to the Cambrian explosion of 13 phyla. All this is propagating, self-constructing organization of non-equilibrium processes into the indefinite diversity and complexity that can arise. This exploration is into what I will call the adjacent possible of the evolving biosphere that evolution itself creates. Evolution creates what can possibly arise next. That I don't have time to really go into it, but it is it's in a couple of my last books. If you read, my couple last books include Humanity in a Creative Universe and A World Beyond Physics. Life creates the opportunities in which it becomes. So does the economy. Once you've got cell phones, the opportunity for apps on cell phones has come into existence. The adjacent possible. The adjacent possible is what can possibly arise next, given what is actual now. Given fish, amphibians are in the adjacent possible of the biosphere. Given Apollo, the space station is in the adjacent possible of the econosphere. All of this arose from the Big Bang, somehow from the Big Bang to New York. So the free energy for this evolution is based on a law of entropy. Well, this is an old theory that you'll find in Lineweaver. Lee Smolin and Carlo Rovelli are advancing different ideas, but let's just look at this right now. It doesn't change the problem quite. So in this view from Lineweaver, the free energy for evolution to occur is based on low entropy state of the early universe and the accelerating expansion of the universe. Thus, the maximum entropy of the universe is growing faster than the actual entropy, so that there's always a free energy available to do thermodynamic work used in the construction, used in the open-ended evolution of the biosphere. So this is Lineweaver. But here's the problem. The availability of free energy is only a necessary condition, not a sufficient condition for the vast emergence and complexity of the universe since the Big Bang. We don't know why the universe got complex so far. Biospheres on Earth, or among the 10 of the 22 solar systems in the universe, literally construct themselves upward into the exploding adjacent possible of diversity and complexity that evolution itself creates. There's no limit upward in complexity. This is indefinite sink upward of complexity. We make skyscrapers now. And hang on, I've just left. Excuse me. Now I'm going to make another radical claim. Neither quantum mechanics or classical physics alone or together explain the evolution of the biosphere. Well, I just said it. Neither quantum mechanics nor classical physics alone or even together explain the evolution of the biosphere. Mutations, say in a Drosophila melanogaster, are random, ontologically indeterminate quantum events occurring in the fly's DNA. These are random events counterfactually changing the DNA code script boundary conditions by which organisms construct themselves as open systems. The random mutations may create a DNA molecule now effectively classical in the fly. The fly itself is effectively classical, rendering the fly with the DNA mutation fitter than before. For example, a mutation that changes a red eye to a white eye. Due to natural selection, soon millions of copies of that DNA molecule, making white eyes rather than red eyes, will exist in the non-organic universe above the level of atoms. So, long other talk, Longo, Monteville, and I in 2012 have demonstrated that no laws entail the evolution of the biosphere. The reason is we cannot prestate the phase space of ever-changing phase space of functions in the open-ended evolution. So we can neither, since we can't state the ever-changing phase space, which is finding new uses of things, we can neither write laws of motion nor integrate the laws of motions we don't have. So no laws entail the evolution of the biosphere. That's a whole long, complicated topic. And it's in my article, Eros and Logos in Angalaki, and my last book. Thus, neither quantum mechanics or classical physics alone or together suffice to account for the specific evolution of the biosphere, which in this sense is beyond entailing law. As noted, the universe is non-organic above the level of atoms. Most complex will never get to exist. So coming into existence in the universe of the mutated DNA molecule in the population of Drosophila melanogaster is unentailed. Quantum and classical physics alone or together fail to account for the existence of this DNA sequence in the universe. There is no final theory. Reductionism stops by itself, stops at the watershed of evolving life. So I'm going to now talk about a tentative effort for a fourth law of thermodynamics, and I think I'm just going to skip ahead to it. The basic idea is that the adjacent possible of the universe tends to increase. The universe grows into the growing adjacent possible it itself creates as it becomes more diverse. This is the first shot at a possible fourth law of thermodynamics. Lee Smolin, Marina Cortez, Andrew Little, and I are within a few weeks or a month of posting online a much better paper about what a fourth law is. And that's for the four of us to get up in about a month. So let me try to summarize this rambling thing. So Schrodinger asks two questions. What's the source of the order in organisms? He says it's an aperiodic crystal. He was brilliantly right. He found the structure of DNA, coding, and so on based on that. He asked the question we still haven't answered. How do organisms cope with the second law? Overall, my answer, I think, is at least either right or a good part of what is right. It is work is the constrained release of energy into a few degrees of freedom. The more the constrained release of energy, the more constrained the release of energies, the more work is done, and the more the production of and release of entropy is delayed. That delayed production of entropy can, for example, radiate it into space, by which the whole planet stays more ordered in the face of the second law. So presumably we can work out how organisms do it as well. But then we have this wonderful notion of constraint closure, where cells do something that automobiles don't do. They construct their own constraints on the release of energy, by which they construct themselves and construct those very same constraints. So cells, unlike automobiles that don't construct their boundary conditions, living organisms do. That's the constraint closure. And now we get to the biosphere constructing itself into an ever-enlarging space of adjacent possible new functionalities that cannot be pre-stated, but there's no law for that becoming. So the trees outside your door and the bugs came to exist without any entailing laws. If you look at my last book, or Eros and Logos makes it clear that all of physics and chemistry is in the framework of the Newtonian paradigm of laws of motion in differential form, initial and boundary conditions that create a pre-stated phase space, then the integration of those equations to yield an entailed trajectory in the pre-stated phase space, like billiard balls on a table. That doesn't work at all in biology, because the phase space of evolution, in biological evolution, includes functions, for example, hearts, or the fact that an elephant in the summer can use its trunk to squirt water on itself and stay cool, and so survive. So the functionality of the elephant's trunk includes that. But we cannot pre-state the ever new functionalities that arise. It's my screwdriver argument. Therefore, reductionism alone stops at the watershed of life, and we need to marry reductionist explanations with functional explanations. The entire becoming is kind of magical. So I've tried to answer Schrodinger's second question. I've not talked about the fact that when a cell constructs a structure like a large protein, that also delays the production and release of entropy until the organism dies, for example, and that protein is metabolized in the soil by some dead bug or some bugs. So two ways that the production of entropy is delayed is it's delayed by the work done, then structures are made that have later to degrade to produce and release entropy. And I have not got that as this article yet. So the big picture is life is just magical. And it probably has started by molecular reproduction in sets of organic molecules everywhere in the universe about 5 billion years ago. So thanks, and I guess that's done. Carlos, over to you.
Thank you very much, Stu. If anyone has questions, please type them down in the YouTube chat, and I will read them along to Stuart. I have a couple of questions, and well, first, thank you for a very interesting talk. We're looking forward to your new draft about the fourth law of thermodynamics. A question would be whether you think there is a difference in information between living systems and non-living systems, or information processing.
I keep being confused, whoever asked the question about information. We all know Shannon. You all know Kolmogorov.
Yes.
For Shannon, you have a source, and there's an entropy of the source, which is the entropy of the numbers of copies of the different messages. And when I get a message at the reception, subject to information lost down the channel, I have a better idea of what the information was at the source. That's fine. You need to know the information density of the source. Now think about evolution. What's the source? There is no source that's being transmitted in life. The whole thing doesn't work. I mean, that's fine. I mean, Shannon was trying to figure out how to send information down a telephone wire. You need a source in his story, and you need something to interpret the source. And of course, that's famously not in information theory. Information theory is about information, but not the meaning. There's no meaning in Shannon. Once you've got organisms, a living protocell, I say, my wife says, and others say, matter, you get from matter to mattering. It's good for me or bad for me. So value enters the universe with Kantian holes. I don't know what to make of information. I keep being confused about it. Does anybody else have any thoughts?
Well, I think that what you mentioned, in a sense, you have already answered, because the meaning of information, following Wittgenstein, we could say that is given in doing.
Yes.
So.
Yeah, Wittgenstein does say that, doesn't he, Carlos?
If you don't consider that, then you cannot get the meaning.
Yeah, that's in his Games of Life. Yeah, Wittgenstein does say that, and he's right. Say we grandly 60 years later, you know, he struggled in the Tractatus with the idea that the world is totally proposition and is a logical construction. Then he gave up on the whole thing, and language games for Wittgenstein are very much the same as organisms finding a way by acting and doing to make a living in their worlds. So the meaning to me of things as say bacterium is this is food and that's poison. The meaning to me as a clam is, a starfish is trying to attack me and I'm going to clam up and I'm going to grow things on my shell so that I'm defended against them in an arm race. The biosphere is absolutely rich with meaning, as millions of kinds of organisms have learned to make livings with one another. And one of the things that I'm finding fascinating is we started out 3.5 billion years ago with a relatively small number of relatively simple organisms. We now are millions and millions of kinds of organisms with millions of copies of each, making livings with one another, and that is the meaning by which we're doing it. My excrement is your food. My surface is something you can crawl on to get food. All that's meaning, right, Carlos?
Yes.
Yeah. By the way, one of the things both of us love, random Boolean nets and criticality and things. That's wonderful. There is no meaning whatsoever in a Boolean net. It's just another nonlinear dynamical system. Once there's a living cell, there's meaning.
Yes, so that makes it difficult to find, let's say, observed independent definition of life, which...
My dog is barking. Sure. Second. Hey, buddy. Buddy. You. Hey. Okay, come on. Okay, buddy. Well, our dog Buddy is guarding us. Some very nice people who are friends have come over, but you can't be too careful. See, Buddy's got meaning out of this whole thing.
Yes.
Let's just take a couple more questions.
Yes. So that leads me to another question, which is related to the detection of extraterrestrial life. That, from what you say, it seems that the only choice is to go there and see it with our own eyes, because all the signatures that we get from telescopes and, let's say, from measuring instruments, it's difficult to get the meaning from those, whether they're organic processes or inorganic processes, whether there's life or not.
Gosh, I don't know. It's not impossible that the complex chemical soups on Enceladus contain molecularly reproducing metabolic, small molecule systems. Well, they can't send messages, but you're right. If we go there, we could see it. There's a guy in Germany named Albrecht Ott who's running the Miller-Urey experiment for a month. And he gets the formation of thousands of kinds of organic molecules. So we can ask in his system, do you get molecularly reproducing autocatalytic sets of small molecules? Those are experiments that can be done on Earth. And in fact, we're trying to get some money from CERN to do them. So we'll see. I take one more question, I think.
Yes, can you make a comment about correlation versus independent systems related to nonlinearity?
Correlations? Ask the question again, Carlos.
Can you make a comment about correlation versus independent systems?
Well, a little bit.
Considering nonlinearity.
Well, you and I both know, Carlos, about order, criticality, and chaos in Boolean networks. Mutual information is a form of correlation. It's maximized at criticality, and yes. Critical systems also maximize an information transfer or a what's the name? Anyway, a transfer entropy. So these are all correlations. And one person who's worked on this is Michele Prokopenko. You must know him, Carlos, right?
Yes, I know him. Michele Prokopenko.
Whoever asked the question. So Michele has looked at information transfer entropy in critical systems. So is Sarah Walker. A group of us in 2007, Roberta, I'll think of it in a second. We established in 2007 that that mutual information T+1 is maximized in critical networks. I think there's a lot of stuff to be done there. Carlos, are we gone? Listen, I need to say goodbye. So thank you all. Carla, would you please send me the link to the YouTube when it's up?
Yes, sorry I got cut off. Thank you very much for your time. It was a fascinating talk.
Thank you, Carlos. Please send me the link to the YouTube. Carlos?
Sure.
Thank you.
Have
Thank you, everybody. Goodbye. I've got a dog to take care of. Bye.
Bye.
Bye, Carlos.